cilia and flagella. Singular cilium and the flagellum. Cilium, there. Hair-like structure. As for example, you found in the paramecium and in the flagellum. This is flagella. This is flagellum found in euglena. And they help in the movement or locomotion, and it is also helpful in the movement of cytoplasmic particles and protoplasmic particles, like that. And so these are the they both are arises from basal body, that is known as kinetochore. Just you have studied little other, that is centrosome or centriole, that is formed. So from it is arises from basal bodies or kinetochore, and it is covered by plasma membrane. It is covered by, just you have seen that. This is the plasma membrane, and these are the endings of the tubules, and here is attached by these are cilia flagellum like structures, and if you cut that, we will see such type of structures having 9 1 2 3 4 5 6 7 8 9 the doublet tubules. Each have two. One is A tubule, next one is B tubule, and combinedly called doublet tubules. And in the center there is also microtubules, subcentral microtubules are present, which is surrounded by a sheath called central sheath, and in that the radial spokes are also present, which are 9 in number. So it is the structure of the cilia of flagella, and it helps in the movement, and that is help in the locomotion, and it also help in the movement of cytoplasmic particles or protoplasmic particles, understand, or small foods in the cytoplasm. Now the next, the important point is nucleus. You know that nucleus is called brain of the cell. It is centrally placed and it controls all the activities of the cells. And it was first discovered by Robert Brown in 1831. In some book you will see 1832. Okay. So, and it was extended basically by Fleming and called chromatin, and it was basically a stent, and then by Fleming and then call it chromatin. However, the cells are maybe uninucleate or multinucleate. The cells are uninucleate or multinucleate. Some contain, generally it is uninucleate, means contain the cells, but in some cells like red blood cells of human, nucleus is absent. In the human RBC, the nucleus, and also in the sieve cell of phloem, in the mature states, the nucleus is absent. So it is also absent in some nucleus and present in all the other cells. Mainly if there is one, that is uninucleate. If there are many, that is multinucleate. Generally the cells are uninucleate. If we draw the structures, nucleus is a double membrane structure that is known as nuclear membrane, and in that you will see some holes for the interconnection of cytoplasm and nucleoplasm. There is some passage is found, that is known as nuclear pole, that is found, and it is about 10 to 20 angstroms, and the outer membrane is connected with mainly endoplasmic reticulum. In that there is a big structure is found, that is known as nucleolus, and the fluid contents of that is called nucleoplasm. The fluid contents of the nucleus is called nucleoplasm. By this passage, there is a connection between the cytoplasm and the nucleoplasm, and in that the thread-like structures is found. A thread-like net is found, that is known as chromatin reticulum. That is called chromatin reticulum. A net-like structure is found. So it is about, so all these nuclear membranes, nucleolus, nucleoplasm and chromatin reticulums, these all are combinedly called nucleus. Mainly 1, 2, 3 and 4. These are the contents of the nucleus, so they are all combinedly called nucleus. Nucleus content. Now we shall see the structure of that chromatin reticulum. Now the chromosome, that is chromatin reticulum. This is the chromosome having chromatids, and the central parts is called centromere, and here is a disc-like structure you get, that is also called kinetochore, and in that filament the DNA, the chain-like structures we get, just right draw a single one. Now this is the chromosome structures, and that a beaded structure is found. This is the main constriction, that is the primary constriction, or that is centromere. And this is the outer membrane, that is known as pellicle. And this is just finger. The tips of the chromosome is just like finger, understand? Not in plane like that, but this finger like rounding. That is known as telomere. It is the secondary constriction. It is also called secondary constriction. Understand? But it is near to the first, so it is known as secondary constriction 1, and it is called secondary constriction 2. And in that, the structure, chain-like structure you see. In that, on this the beaded-like structure is found, and they are called chromomere, and the space between two is called interchromomere. That is the space between two chromosomes, and sometimes the ends of the chromosome after the secondary constriction at once become rounded. That is called satellite. That is called satellite, and the chromosome containing satellite is called sat chromosome. So the satellite containing chromosome is called sat chromosome. So this is the structure of a chromosomes, and it is the centromere. And the position of centromere, on the basis of position of centromere, the chromosomes may be of following type. On the basis of chromosome, that is chromosome, suppose the centrosome may present in the middle, it's above the middle, understand? And that is the last, or at the tip, whatever. In that, if it is found in the middle, somewhat below the middle, that is towards the end side and at the final point. If it is found in the, if the centrum, there is a centromere. Similarly, these two are. If the centromere is found in the middle, centromere is found in the middle, that is primary constriction is found in the middle, that is known as metacentric. It is known as metacentric. It is found in the middle, that is metacentric. If it is found below the middle, that is known as submetacentric. If it is below the middle, that is submetacentric. If it towards the end side, not at the end point, but towards the end, that is known as acrocentric. And just at the point tip, it is known as telocentric. Understand? And you know, on the chromosomes, the DNA is present, and DNA is the genetic material. Understand? It transfers the characters from parents to their offsprings. That is the, and it is found in the nucleus.